Hi, I'm Maria Nattestad, and I'm going to tell you a little bit about SplitThreader. SplitThreader is a visualization and analysis tool for long-range variants and copy number. It's useful for looking at genomic rearrangements and copy number amplifications, especially in highly rearranged cancer genomes. It takes as input a copy number profile and a set of variant calls, and it's at splitthreader.com. So I'm going to show you a quick view of what SplitThreader looks like and what it can do for you. So let's hop on over to splitthreader.com. So here's splitthreader.com, and you can see there's instructions on the right side that tell you how to generate your variant calls and your copycat copy number profile. So all we have to do is enter a set of variant calls. I have here a bedPE file from Sniffles, which is generated using long-read sequencing data, and a copy number profile. And I'm going to enter a quick little description of what we're doing here. This is SKBR3, which is a breast cancer cell line, and it's using Sniffles calls. So I just wait real quick for it to upload. That was all. And we submit it. And it says both files were uploaded, so that's awesome. We click continue. And it has already parsed the variant input file. It even filtered out a bunch of the variants that are too small. So the minimum we have in SplitThreader is 1 KB. Any variant smaller than that, you're better off using IGV, because SplitThreader is really about the long-range rearrangements, and it's built as a way of visualizing those that IGV doesn't have much of a hope of really displaying and showing how all of the different regions in the genome are going to be connected once these rearrangements have taken place. So while we wait for the copy number segmentation to finish, which only takes less than 5 minutes usually, under the Info tab, you can see how to cite our paper and how to get the code on GitHub. My data will tell you all of the call sets that you have already started running yourself. Oh, our run actually finished already, so let's click on Show Visualizer. And it just takes a second to pop up. Here you can see a Circos plot of all of the variants that are more than 1 KB in size, remember that. And all we have to do here is take a look at the copy number profiles. These are segmented, and it shows by default chromosome 1 and chromosome 2. SplitThreader works for human data. It would also work on mouse or anywhere else that you might be studying various things, but I recommend mostly using SplitThreader for cancer genomes. So to navigate around SplitThreader, you can drag out a chromosome from the Circos plot onto each of the copy number profiles here, and we're going to switch to that chromosome. If I drag out chromosome 8 here, you can now see all the connections that go across here are connections between chromosome 17 up at the top and chromosome 8 at the bottom. You can double-click to zoom in on a region you're interested in. You can also use the plus and minus buttons up here to zoom in and to zoom out. And as you do that, the connections you're looking at are going to adjust as well. So if I zoom in on one of these locations, you can see some of these connections went away because the regions they're connecting to are no longer within view. But if I zoom back out, they'll show up again. They stay there as stubs, so you can see they're there, and you'll just know that they connect to this other chromosome based on the color of the variants. You can also zoom in on a particular variant by just clicking on it. You can see it gets bold when you hover over it, and that way you can see what variant you're looking at. You can search for genes. If I type in MYC here and hit enter, it's going to go straight to where MYC is in the genome in the top plot. And so I can see that MYC is next to another little deletion here of 45 KB, which is a deletion. And in SR right there, it says 159, so that means it's supported by 159 split reads. And there I show whatever information I find in the VCF file when I parse that. So it's all dependent on what the tools you're using are actually going to be showing in the VCF file. That's an important point. We have examples up here that you can take a look at. There's SKBR3, MCF7, and A549, all of which are covered in our paper that we just put out on the bioRxiv. You can also go to the variant analysis tab to look at different categories of variants, to do filtering. You can type in here to say anything like, I want any variant where the size is greater than 10 KB, so you go greater than 10,000 and you hit enter, and it's going to filter this table. It even updates the chart up here that gives you a variant size histogram, and you can then also see the number of variants here out of how many there are total. This categorization matrix, which I describe in detail in another video, consists of a variant neighborhood category affected only by how close variants are to other variants and how they're located relative to other variants in general. And then the copy number concordance category has something to do with the particular variant that you focus on. Does it have support from copy number changes that match with that variant? Another thing you can do is you can type in gene fusions. And so you just type in 2 names of genes that you think might be fused, either from previous literature or from your own RNA-seq experiments or something like that. I choose to upload a list. So I just have a list here that has 2 different gene names in column 1 and column 2, which can be separated by tabs, commas, or spaces. And then SplitThreader goes and it searches through a graph of rearrangements to find the shortest path between each of these sets of genes. And what we find here, for instance, is that we have a connection. Let's pick one of my favorites, Sith1EIF3H. This is a really cool one because it's one of these 2-hop gene fusions. So when I zoom out a little bit, I see that there's 2 variants involved here. One that goes from Sith1 into the sequence on chromosome 8, and then it runs for, it showed me before, it was about 8 KB to the right, and then it continues to the left. And if you want to see what that looks like, you can zoom in on any one of these points to just get a lot more detail. You can use the plus as well. And if I just keep zooming in, I can see that the variants are very close together here. So that's just a small taste of some of the stuff we can do in SplitThreader. Note that you can also export the regions to the UCSC genome browser just by clicking on these links. And you can send variants to Ribbon. You can send your gene fusion variants specifically to Ribbon, which is pretty cool. So it'll take all of these and you just click a single button and it'll open up genomeribbon.com and put those in as rearrangements as if you had uploaded a BedPE file where you had specifically extracted the variants that have to do with these gene fusions. So you can go and investigate for each gene fusion, what do the alignments look like in Ribbon around those variants. Thanks for watching, and please let me know in the comments if you have any questions or any comments about SplitThreader. I look forward to hearing from you. Thanks!